All right, welcome back. I'm delighted to begin our second session where Dalya Barron and Adam Bolton are going to tell us how the universe works, I guess. Take it away.
In 15 minutes, yeah. Hi, everyone. I'm very excited and happy to be here and discuss with you all the interesting research opportunities we have in common. And yes, we're going to sort of dive into what is the physics of astrophysical objects that we're interested in. And for that, we first need to understand what do we have in the observable universe. So what you're seeing here is an artist illustration, is not real, of everything that is in our observable universe. We're centered here, the sun, Earth. We have galaxies, etc., as we will discuss, and all the way to the cosmic microwave background and to the edge of what we can see with our telescopes. Now, again, this is an illustration, so the relative size of things and the distances of things from us is not to scale, but it sort of gives you an impression of what is it we're interested in as astronomers and astrophysicists. So within our solar system, we obviously have our own planets, and we have known for the past 2 decades, 3 decades, of the existence of extrasolar planets, so planets orbiting other suns, other stars in the Milky Way. Within our own Milky Way, we have stars and we have compact objects like neutron stars and black holes, where the surface gravity is much stronger than that of our own sun. We have the gas inside the galaxy, like what Susan described, and we have the gas outside the galaxy in the dark matter halo, called the CGM. We have local and distant galaxies that we can see. We have galaxy clusters. In these distant galaxies, there exist supermassive black holes that sometimes accrete. Their mass is between millions to billions the mass of our own sun. And when they accrete, they emit radiation throughout the electromagnetic spectrum that we can see, quite bright. And in addition to that, we have different transient phenomena, so things that go bright and then dim in the night sky, like supernovae, for example, which are exploding stars. And we're interested in understanding all of these sources.
So how do we actually understand all of these things? How do we make sense of them? As good post-Copernican scientists, we make the reasonably well-grounded assumption that the same physics governs what happens out there as happens down here. And applying that physics is what we call astrophysics, and it's how we understand the physics of astrophysical or astronomical objects. Of course, it's a whole field of study that we're trying to boil down into a few slides here. So I'll pick a particular relation to feature that I think has more explanatory power than any other single relationship. So it's the same physics out there as down here, but of course the energy scales, the mass scales, the length scales, the times are vastly different than what we're used to dealing with. So we deal with conditions and phenomena that are very much beyond our local experience. And this fundamentally, in large part, comes down to the interplay between things like thermodynamics and quantum mechanics and electricity and magnetism that operate locally, and gravity, which operates globally over very large distances and essentially attracts everything in the universe to everything else. So if you roughly define an astronomical object as a self-gravitating bound system, and you apply statistical mechanics of a sort to that system, assuming it's in something resembling equilibrium, you get a relationship that the kinetic energy is equal to 1/2 the negative of the potential energy. This is a version of what we call the virial theorem. There's a few things to note. One is that astronomical objects have negative heat capacity, so the more they cool down, the more they heat up, which, figure that one out, but okay, that's interesting stuff. Also, you'll see that the kinetic energy of any particular system, roughly speaking, again, is proportional to its mass and to the inverse of its radius. And so if you apply this to things like, say, putting the mass of the star in something about the size of the star, you'll find out that the temperature in the center is somewhere in the neighborhood of 10 million Kelvin or Celsius, which is hot enough to force protons to fuse together and liberate energy through nuclear fusion, which can counteract any further contraction of the star and have it live longer and give rise to life on planets like Earth. If you look at the kinetic energy of the orbital kinetic energy of stars in galaxies, or of galaxies in entire clusters of galaxies, you'll find that there's more mass than you can account for just by their stars alone. That's how we know of dark matter and why we think it's such an important part of what the universe is made of. And then going back to those stars, once they use up their nuclear fuel and contract down further to the point where their size is regulated by quantum mechanical effects of the electrons, you find that they get down to be the size of the Earth with something like the mass of our sun. And if you add even a little bit more mass, the electrons go relativistic and the whole thing collapses and then explodes as what we know as a type 1a supernova. So you can do this for other sorts of objects as well, but just moving on, since we don't have too much time, I think the other important piece of astrophysics to take towards trying to get a general intuition for what's going on is if you combine electromagnetism and quantum mechanics and statistical mechanics, you also learn this lesson of the black body spectrum, which many of you are perhaps familiar with. The things to take away from this are that the total luminosity of an object increases with its temperature for a fixed size. Colors also get bluer as temperature increases. And then, as we've already seen in some of the previous presentations, atomic transitions complicate this simple picture, and they give rise to more details that we can observe in these simple distributions here.
So integrating everything that Adam said, we realized that astronomical objects emit radiation throughout the electromagnetic spectrum. And here I'm showing you a representation of the electromagnetic spectrum from radio waves through microwave, infrared, visible, so what we see with our eyes, UV, X-ray, and gamma ray. And astronomers have been very productive in trying to map the entire electromagnetic spectrum, giving rise to different points of view for the same astronomical source. And as Susan explained, we have several data products we work with. One of them is images in these different wavelengths that give us a different type of point of view. We have spectra, which is an intensity, so amount of light as a function of the wavelength, and we also have light curves, so intensity as a function of time. And these are the three basic observables we use in order to study the physics that govern the astronomical objects we're interested in. And the sort of something that should open this quarterly meeting, I think, is the fact that astronomy has experienced a data revolution roughly 30 or 40 years ago. So before that, science was done when we began with a hypothesis, and we designed an experiment, a set of observations to test this hypothesis. But since then, the dawn of astronomical survey started, where basically we entered into a survey mode where we have these huge surveys observing large number of sources in the night sky and providing this data in a very convenient form for all astronomers and scientists. And first of all, it means that you no longer need to have a telescope to do science. And this was very revolutionary at the time. In addition to that, you can see that so the data volume and the number of observed sources increased exponentially. We're having this very robust and large statistical population of different sources, whether galaxies, stars, supernovae, to study. And the thing that I find most exciting is the fact that all of these different surveys covering different portions of the wavelength range cover the same astronomical object. So now the dimensionality of each astronomical object is huge. Now, in computer science, we're talking about the curse of dimensionality. This is something that we used to say in computer science. I want to call this the blessing of dimensionality, because within these different dimensions, there are these new relations and discoveries that can be made. And this led to the dawn of data science in astronomy. How can we use the data from all of these surveys to make new discoveries?
And so to try to cast the data challenge and the sorts of data analysis approaches that we bring to these very large survey data sets in the most perhaps simplistic, certainly oversimplistic way possible, we can think about it essentially as a problem of going from the images and spectra that we have to particular observables, ideally physically meaningful observables that we can extract from those images and spectra. And then we can do that for populations of millions or billions of objects. And to look at it in a sort of a schematic, cartoonish way here, to plot some points and see that there's some sort of population here. We want to be able to characterize those object populations. Maybe these are stars, maybe these are galaxies, maybe we're observing their luminosity, maybe it's their distance, maybe it's their internal velocity dispersion. All these different measurements of empirical relationships, scaling relations between different properties encode things about the formation of galaxies, things about the evolution of stars or populations of stars or galaxies. So we're looking to characterize these things from these many millions or billions of data points. We're also looking to detect anomalies and outliers, interesting rare objects that don't fit with the main population. And of course, we're looking for effective and informative ways of comparing the predictions of either predictive or descriptive theories to these very large data sets. So that's the kind of the ideal version of it, but it does, of course, things get more complicated. One thing that's, I think, really important, certainly it's been salient for me looking at how recent work in machine learning either does or does not translate into astronomy, is that I see a lot of machine learning applications, and I think that's great. That's infinite signal to noise. Wouldn't that be great? In a lot of astronomy and astrophysics, we're working very deep into the low signal to noise regime. We also have variable errors, or I guess heteroscedastic is the snobby way to say it, I guess. So yeah, all the data points have different error bars, and they have different error bar if you go back tomorrow as they do today. We have missing and irregularly sampled data. And of course, as Dalya already mentioned, these data sets have now grown to the petabyte scale. They're much larger than anybody individually can download and analyze on their laptop. So we have to use not only shared infrastructure for doing the observations, we actually have to use shared infrastructure for doing the data analysis. There's other things going on. We often have selection effects that are not independent of the physical observables that we're trying to constrain. And then we almost always have very complex and very often oversimplified relationships between the experimental design and the apparatus versus how that flows down to the more idealized data products that we ultimately work with.
So just to finish, we wanted to give you two examples of surveys that have been happening and are going to happen in astronomy that we're very excited about. And the first is the Sloan Digital Sky Survey, which in some sense pioneered this data revolution I was describing. It started in the 2000s, and it's now in its fifth phase. And during the survey mapped roughly 30% of the night sky. And you can see here the numbers. They're astonishing. So there are about 10 to the 9 images of stars and galaxies, 10 to the 6 spectra of galaxies and stars in the optical, 600,000 of infrared spectra of stars, 10,000 galaxies with both imaging and spectra. So these are now cubes of data. And I think to sort of understand what is the impact of this survey in astronomy, I'm citing here roughly the number of papers that used SDSS data. And I think this gives you a sense of how revolutionary it is in astronomy.
And then just to bring us now into the present and the near future, the latest and greatest version of experiment, of survey, of observatory for big data astronomy that's being commissioned even now as we speak is the Rubin Observatory, which will conduct the 10-year legacy survey of space and time starting sometime in the next calendar year. This is named for Vera C. Rubin, who back in the 70s provided the first evidence for the presence and the spatial distribution of dark matter in other galaxies. So this project is going to use an 8.4 meter telescope. It's on the summit of Cerro Pachon, which is in northern Chile. You see a picture of it in almost completed form there from the outside. It's going to image half the entire sky, well, essentially the entire visible sky from its southern hemisphere location every 3 to 4 days to build a 10-year time series database of something on the order of 20 billion stars and 20 billion galaxies, and approaching a sort of 100 petabyte scale data set by the end. So this is something where SLAC has played a leading role. It's the lead lab for the DOE side of this experiment. It's also, SLAC built the camera, we host the data facility, we also host the Dark Energy Science Collaboration. And so this is, again, this is a collaboration between the NSF and the Department of Energy, which really highlights the way in which data-driven, data-intensive astronomy and astrophysics has made common cause with the cosmic frontier of high-energy physics in this era of wide-field surveys and giant data sets. So this is coming now, and a bunch of us are really excited about it. Some of us are stressed out about it, but that's just how it goes. That's part of what makes it fun. So. Yeah, I think we can leave it there.
Awesome. Questions for Adam and Dalya? Let's see. Maybe I would love to hear what each of you are most excited about with LSST data imminent. Excited, not stressed.
First. So I'm interested in it ultimately being a targeting survey for an even larger spectroscopic survey 10 or 20 years from now. I know that sounds like too far off, but that's my honest answer.
I think that I'm most interested and excited about the deep LSST images that will come after we're going to stack all the individual exposures of galaxies. It's sort of revealing the structure around galaxies. So this is something that I think that will be very interesting. Risa mentioned stellar streams around our own Milky Way and the detection of stars in these streams. This is something that unfortunately can be done only in the Milky Way or in very local systems, because it's very difficult to detect individual stars in other galaxies. With LSST data, the number of structures around galaxies will grow, and I think that it will open up some new parameter space to explore and study in the context of accretion and dark matter.
Maybe, oh, sorry. I was going to amend my answer too, because I had a better one, but go ahead.
So, Adam, you didn't mention that part of your job is to deliver the million things that change every night.
Yes.
And very quickly. And I guess I'm wondering, like, I know you have some plans for this, and the community already has some plans for how they're going to deal with this, but I'm curious what your current thinking is on, like, how much that can and should change in the next few years with different modern techniques.
Yeah, so what Risa's mentioning is a part of the LSST that I didn't describe in too much detail, which is that not only are we building up a movie that we can go play back, we're actually going to be watching it in real time, and if something exciting happens, we're going to find out within like a minute of the shutter closing and potentially trigger other follow-up observing. So that's done right now through a fairly well-defined and well-established way of doing difference imaging relative to templates and broadcasting alerts out. And then people can deploy various filters or classifiers on that alert stream. The estimate is that there'll be something like 10 million alerts per night, so it really does need some kind of automation to winnow that down. Where I think there's a lot of opportunity eventually is being able to deploy trained models, more sophisticated trained models closer down to the source of the alerts, like maybe even right in the data center where we're broadcasting these things from. That's beyond the current scope of the project, but thinking a few years into the future, I don't think there's any reason why we couldn't look for new ways of doing that alert processing and bringing the AI and the machine learning closer to the raw data and to the real-time processing. It's actually interesting, has interesting resonance with the way that, as I understand it, that they're applying machine learning and AI to accelerator beam control in the other parts of the lab. So we could imagine similar sorts of thinking going into the alert processing as well.
Awesome. All right, let's thank our speakers again.
All right. Hi, everyone. Can folks hear me okay? I'm going to ask folks to allow me to change gears, change topics to biology, where we apply foundation models to biology. And hopefully it's a useful use case to see how we partner up with a machine learning lab and a biology team, another area of science, right? My name is Eric. I'm a PhD student in bioengineering, and I'm really excited to share our work today on a foundation model for DNA and genomics. The model is called EVO. It's a recently accepted paper into Science, so we're really excited to share and give a preview to folks. And so it's just a preview, be fairly brief. And so if you find yourself wanting more, great. This project is all about treating DNA as a language. And so if you think of yourself as a book of genetic code, this book has 23 chapters made up of chromosomes, is made up of about 3 billion characters or nucleotides with just a simple vocabulary of 4 letters. Now this analogy to language for DNA is appealing because, as we've seen, machine learning and AI is very good at language, right? And natural language in particular. And when you train it with massive amounts of data, like the internet, you get cool applications like ChatGPT. The great thing about DNA is that it's one of the most abundant sources of biological information. And it's growing. The cost of sequencing and getting more genomic data is rapidly, is really fast. So it's favorable in terms of the amount of data. Now, why would you care about trying to aggregate and learn from all this amounts of data? Well, it can help with understanding disease better, potentially, drug discovery, and also potentially help with this concept of personalized medicine. Now, there's a long way to go before we can get to such cool and impactful applications. And so for this work, we had to target something that was both exciting and achievable, which I thought was really captured very well by this quote that said, "Today we can, for all practical purposes, read, write, and edit any sequence of DNA, but we cannot compose it." And this was said by Frances Arnold, Nobel Prize winner in chemistry. And what she was getting at was that we don't understand enough about DNA to design it. And so that's when a group of researchers in Hazy Research, machine learning CS, computer science lab at Stanford, teamed up with the ARC Institute, a new biology research institute across the street. And one of the things we asked was, can we design an entire genome from scratch using AI? To do such, or to pursue such a bizarre task, we had to put together a data set. All right. And so we put together one of the largest known data sets at the time, and we called it Open Genome. It's made up of a bunch of raw genome sequences from prokaryotes, essentially microbial life. And then we started training a foundation model. So if you're unfamiliar with how foundation models are trained, you simply sample a sequence from this data set, and then you essentially predict what's the next letter or sequence, next token in this sequence. And you do this billions of times, and you hopefully come up with a model that's understand the distribution and underlying statistics of your data. And then you can apply it to downstream tasks that you actually care about. We have a host of tasks that I can only preview, unfortunately, just here, from prediction tasks, essentially correlation tasks, to generative tasks, which I'll share a little bit about. So one of the interesting tasks that we're really excited about are CRISPR-Cas systems. So there's a family of gene editing tools that are mined from nature, and so we wanted to see, can we generate one from scratch using AI? And so what you're seeing here is one of the first AI-generated CRISPR-Cas systems, synthetically, and that actually work in the lab. And so these have been validated in the wet lab, which is what we're also really excited about. Similarly, we also synthesized transposons, which is also another family of gene editing tools that works differently than CRISPR-Cas. Essentially, it can allow potentially larger edits to DNA. And I just want to close that preview with just mentioning that this was a very large disciplinary team of machine learning folks, computational biologists, and wet lab biologists. And I want to point to some references that you can find more information if you're interested, where we can talk about the paper itself, the code base, which is public. We make the weights to the pre-trained model entirely public, and so we're really excited about folks and seeing what they are able to build on top of. I'll close there, and happy to take any questions, and also my email is there if you have further questions.
Awesome. Phil.
This is more a question for someone who hasn't taken biology in 14 years.
Beautiful. Same here.
So could you just remind someone like me what the connection between DNA and protein folding is and these sorts of things? I remember that they're in the same regime, but not exactly what the connection is.
Oh, sure. So for protein folding, people care about understanding how these sequences are formed in a three-dimensional environment because it influences their function. And so in our case, we're sort of agnostic to the structure at this point. The hope is that just through learning from the sequence itself, the sequence space, that we could glean some of the structural information or maybe even bypass it so that we can just generate sequences, for example, sample novel gene editing tools, without needing to explicitly predict the structure, but sort of just search the space of potential sequences. So yeah, I think that's one way to look at it.
Thank you.
Saria.
Yeah, very cool work. I was curious, how different are the generated samples that transpose onto CRISPR-Cas systems from stuff that's in your training set? Like, how creative is the model?
Yeah, this is a really good question in terms of sort of diversity from the training set. Can you really say these are AI generated? Not a simple answer necessarily, but one metric we use to evaluate how different these AI generated gene editing tools are from those in nature. We use this metric called sequence identity. Essentially, for every position, how much is it exactly the same as those in nature or those in training set? And so it's a percentage, so 0 to 100. And we basically chose a distribution, so some that were something along the lines between 40 to 80% similar. And so we have ranges of some generated from like low similarity to high similarity. And we have ones that work, I guess, along the spectrum.
One more
Do we have
from
time?
Agnes while our next speaker sets up.
Thank you. That's really fascinating. And
Thank you.
I feel like it must also pose some ethical question as well.
Absolutely.
And my question is more about what do you mean by you tested this AI generated things in the lab? Like what are you actually doing?
Yeah. Good question. So essentially, we're able to take the design of the model, so it outputs a sequence of DNA characters. We could then essentially send it to a lab where they'll synthesize the DNA. So they'll essentially put together the links of DNA, and then we could retrieve those and then express them, meaning we can convert that DNA as a blueprint and make the proteins or gene editing tools in the lab and synthesize them in vitro.
All right, let's thank Eric again.
Thank you.
Okay, so today I'll tell you a little bit about a line of work that I've been working on over the past couple of years that's called prediction-powered inference. Also, my name is Tijana. I'm a postdoc here with Stanford Data Science and the Department of Statistics. Okay, so I'll be talking about a few different works. And so all of these are sort of the main references that I'll be talking about. And these are joint works with Anastasios Angelopoulos, Stephen Bates, Emmanuel Candes, John Ducci, Clara Fan-Jiang, and Mike Jordan. So these are all collaborators from Berkeley and Stanford. Okay, so the motivation for this line of work is basically that we're seeing an increasing number of settings where machine learning predictions are replacing real data. So for example, this is something that we're seeing in policy, where predictions from satellite imagery are replacing survey data and ground-based measurements. So in particular, what people are doing in remote sensing is they're taking satellite images from readily available platforms, such as the Google Earth Engine, and they are predicting different kinds of socioeconomic or environmental indicators from these images. So for example, they're training computer vision algorithms to predict things like population density, deforestation, poverty, and so on. And these computer vision predictions are a much more efficient way of measuring these phenomena than more traditional methods, such as sending trucks out into the Amazon to measure deforestation or conducting expensive surveys on the ground. This is certainly something that's happening in science, so this relates back to the previous talk. So we all know the excitement that the release of AlphaFold generated, and after this Nobel Prize from a couple weeks ago, it's only going to generate more excitement. So basically what happened was in 2020, DeepMind released this model called AlphaFold that predicts a protein's 3D structure from its amino acid sequence. And this generated a whole explosion of new biology research, because prior to AlphaFold, we had only on the order of about 200,000 experimentally derived protein structures. But now after we trained AlphaFold, all of a sudden we had protein structures for just all proteins that are known to science. And so of course, this abundance of new data generated a lot of new scientific questions and a lot of new research. So what we can see in these examples is that there is a pretty good reason for using machine learning predictions as a substitute for real data. In particular, machine learning predictions are typically abundant. They are generally cheap to compute. They're fast to compute. Real data takes a lot of time to collect. It's very, very expensive. In the AlphaFold example, it requires very expensive experimental techniques. It's also very slow to collect. But of course, there's a flip side, which is that what we typically call real data is basically gold standard measurements of whatever phenomenon we care about measuring, whereas with machine learning predictions, we typically don't really have any guarantee on what we're actually getting. So, of course, as a result, any kind of downstream conclusion that just naively trusts these machine learning predictions has to necessarily be invalid. And so that brings me to the topic of our work, which is a general protocol for drawing valid conclusions based on machine learning predictions. This protocol is pretty general. In particular, it can be applied with any black box machine learning algorithm, which means that it can be applied to both of the previous settings that I described. It comes with provable validity guarantees. And finally, it is applicable to a pretty broad class of estimation problems. Okay, so just to very briefly go over the setup that we're working with. So we are assuming that we have capital N unlabeled data points drawn IID from some distribution, and then we have a smaller set of little n labeled data points drawn from the same distribution. So here we're observing these x and x tildes and y, but we're not observing this y tilde. And really the setting that you should keep in mind is that this capital N is much, much larger than little n. Okay, and again, if you want to have a running example in mind, you can think of these x's as being satellite images. So we have a lot of satellite images, but we have only a smaller number of images that have actually gold standard annotations of, say, the deforestation rate. Okay, and so in addition to this data, we have a good machine learning model that we don't know much about its accuracy, but we suspect that it's pretty good. And of course, intuitively, what we would want to do is we would want to take this machine learning model and just impute all of these labels that we don't have. And so what our method does at a super, super high level is, given a quantity of interest, it first computes a so-called naive estimate, where it treats these machine learning predictions as just being true labels. And then it subtracts off an estimate of the prediction bias as estimated on the small label data set. And so we can do that on the small label data set because there we have all the information. Okay, and the guarantee is that it will produce a confidence interval that has the right coverage. So for example, 90%. Okay, and so I don't really have time to describe all the applications that we've looked at. Here, I just want to give you a bit of a sense of just how much you can gain from using these machine learning predictions as opposed to just standard statistical techniques. So here I'm showing you 3 different applications with very different data modalities. And so here you can see that over just classical approaches in statistics, we can really gain even over 75 or 80% in our sample size. So the gains can actually be quite significant because these models have very, very good predictive performance. Anyway, that's all I wanted to talk about. I'll just say that we do have a package that we worked a lot on, and it has pretty detailed documentation. So if this sounds like it could be interesting, please do look at the package. Yeah, thank you.
Fantastic. Questions? I guess.
That's very interesting, and I can think about an application for cosmology.
I would love to talk more.
Yeah, because we can, like I imagine, we can use like generative AI to create like some simulations that are very expensive. But I'm wondering what is like the percentage of data that would be acceptable to have as predicted by machine learning compared to like real simulations, for instance?
Yeah, so yeah, that's a good question. So it really is important that we have some unlabeled data that is like true information, and then we're just generating data from this true information. So for example, in the AlphaFold example, we are assuming that we have amino acid sequences that we care about, and then we are just generating the protein structure. So there has to be some initial input to the procedure that you know is in some sense valid. I'm happy to talk more offline, but I think that's a very good question, and that is also like a direction that I'm actively thinking about right now. Yeah. I don't know if we have time.
We've got
So,
one
one
more.
more, okay.
How much does your framework care about the fact that black box is a machine learning model? It's like if it's a complex ODE, someone's polynomial fit to something, does it care about that?
No, it doesn't care about that at all. So yeah, it can really be black box. That's why we're kind of emphasizing black box, because it can really be anything. Yeah. Thank you.
Fantastic. Thank you.
Thanks so much.
Yeah, hi, I'm Sydney. I am a fourth-year grad student in CAIPAC, and I'm also a Stanford data science scholar. And yeah, I just wanted to introduce, there's Dalya and Philip who are down here, and then also Joe, who is coming in person soon, and some other people as well. We've been thinking about starting a journal club in the center, and so we're all really excited to invite all of you to join us there. We're going to be meeting every other Monday at 1:30, and then once a month we'll do a 1 p.m. lunch before that. And so yeah, our first meeting will be on Monday, November 4th. It'll be somewhere in PAB. We'll have Zoom support, but we want to encourage in-person attendance. And we're aiming to go through roughly 2 papers per week. We want to keep this Astro ML focused, and really focused on methods and concepts rather than like new science results. And then once a month when we do the lunch meeting, we'll have kind of a special format where we'll kind of talk about ideas that we want to discuss at lunch, and then we'll do a whiteboard-only session where we'll kind of like go through some of the ideas that came up at lunch and discuss them further. So yeah, please feel free to contact any of the 3 of us or Joe if you want to learn more, and please come join us. Yeah, thanks. That's it.
Okay, thanks. I'm Gordon Wetzstein. I'm an associate professor in electrical engineering. I work on inverse problems in the context of computational imaging. At least that's what I've been doing for the last 20 years or so. And in the last couple of years, my group has gotten really excited about generative modeling, in part inspired by Surya's seminal work and others here at Stanford. So with this talk, I'm going to try to bring these together, and my hope is that I'll find some good applications and problems that our methods can help solve in this context. So, I've given a couple talks in this context. Maybe not all of you were here. But one of the motivating factors that I like to use as an example is the mantis shrimp. The mantis shrimp is a creature that lives underwater, tropical waters. Some species are reported to have up to 16 different types of photoreceptors in their retina. Remember, humans have 4, 3 for color, 1 for night vision. So some mantis shrimps can see 12 different spectral channels and also polarization, so that's incredible. They have a very different eye. The resolution certainly isn't as high. But the idea is that it's probably evolved in its specific environment and optimized for perhaps survival or tasks in that environment. Now, let's take a look at the vision system of the mantis shrimp. I mean, obviously there are the eyes that are obvious, but then there's also the brain. And the way, at least at a first order approximation, I think about it is that there's some sensing and low-level computing perhaps going on in the retina and in the eyes, and then there's some amount of perception and higher level, perhaps cognition going on in the brain. And these two components have probably co-evolved. So if we translate this idea to our field, inverse problems, then typically what we do is we take observations with some instrument. I mean, in your field, maybe that's a telescope or some other type of an instrument. And the image formation model knows about the physical process of taking these measurements. So there's an unknown target signal, or at least in the context of the inverse problem, it's unknown, but perhaps it's known in some simulation context. There's a measurement operator that defines how is this signal being projected onto my detector, and there's some noise. It could be signal dependent or not, and then there's going to be some noisy and corrupt observations. So oftentimes, the problem of perception reduces to a reconstruction problem. So reconstruction means you want to find the unknown target signal and estimate that, given the measurements, right? So what we're designing here is the reconstruction operator. It could be an algorithm, could be a neural network, could be some other type of a thing. Given the noisy measurements, given the physical image formation model, which is typically known or can be approximated, given some statistics of the noise, a statistical model, but also often we use priors in this context. So prior is some prior information that we have about the signal we're looking for, and that can be something that's handcrafted or learned. And what we get out is an estimated signal. So this is a classical inverse problem. So the questions we try to answer with these types of reconstruction problems is, how do we design the best reconstruction algorithm for some type of an instrument? Are we going to use maximum likelihood, maximum a posteriori? More recently, we've been working a lot with generative models, such as diffusion models, perhaps, for pixel data. And then we use things like posterior sampling, or we use a neural network to approximate this operator to make it really fast. So there are many different choices of algorithms that can help solve this reconstruction problem. We can also ask questions about how to best calibrate an instrument. Let's say you have a complicated multi-telescope setup, and you want to sort of model how the signal actually maps to your measurement, and not all of these parameters are known, then might have to do things like point-spread function engineering or point-spread function fitting. We might have to use AI techniques to calibrate this instrument to get to this operator. We ask questions about modeling the noise statistics. Is it Gaussian noise? Is it Poisson noise? Is it some mix of the noise? Or is it something radically different that we need to model? The more we can model it, the better our reconstructions typically are. And then I would say the most important question perhaps in this case, or the most active in terms of publications, has been, how do we choose a good prior for the model that we have? Because most of these other questions have been, there are good methods that do all of these, but what is a good prior for my problem? One thing that I often see is when I look at inverse papers who solve inverse problems, they just throw a TV prior at the problem without really knowing what that is or what assumptions it makes. But can we learn a data-driven prior, for example, in the form of a CNN, or can we use a pre-trained large text-to-image generation model to fill in missing parts, right? So these are a lot of the questions that we ask, and then you probably know a lot of the applications, and I hope to find more. So imaging black holes, that was a MAP approach, a maximum a posteriori reconstruction of the black hole with a fairly complicated image formation model, but that's an inverse problem. Image restoration and computational photography is one. So the pictures that your cell phone takes, that's an inverse problem. There are noisy observations, especially in low light conditions. When you take selfies with your family in the dark, you want to see a nice photograph. The pixels that you look at in the final image are not the ones that your sensor measured. Perhaps structure for motion in computer vision, medical imaging. Think about MRI, CT. We take measurements in some other domain. They're noisy. They have missing information. How do we reconstruct a 3D volume? Phase retrieval in the context of SLAC, crystallography, also astronomy, optical imaging, and then cryo-electron microscopy, for example. I just want to highlight one project that we worked on in this space. So we've worked on many different projects, but cryo-electron microscopy is a very interesting one because it's a interesting topic, lots of discoveries to be made, and it's not a trivial inverse problem. For those of you who don't know what cryo-electron microscopy is, there are different flavors of this. We mainly work on the single particle imaging case, in which case we have the instrument, that's the electron microscope. We have molecules that are flash frozen in ice. We expose that with an electron beam, and there's a detector that measures the projection of the 3D volume. And we're going to make the assumption that we're going to have lots of different molecules that are very similar. They're almost the same. Maybe they are slightly differently folded. They have some conformational landscape. And then what we measure is a micrograph. So this is a very noisy image with many different copies of this protein or of this molecule, and we can crop out small crops that show a single particle, so a single macromolecule, for example. But the problem here is that, first of all, it's super noisy. If I asked you what it is, you wouldn't be able to tell me because it's so noisy, right? Like, there's more noise than signal here. So that's challenging. There are also, not only tens or hundreds of thousands of measurements of the same object, but millions, tens of millions of these small, low-resolution, noisy images of the same structure that we use as input. And then the worst part of this is that the orientation of these particles is unknown. So yes, we have lots of images of approximately the same molecule, but they're all in different rotations, and it's a 3D structure. So it's not straightforward. We basically have millions or tens of millions of measurements with some unknowns in the measurement operator. And so the default solution for that would be something like expectation maximization. So if you go to the textbook example of how to solve such a problem, would be expectation maximization. It's just not feasible to run at this scale. So now we need to be clever, and we use modern techniques from AI. We build on an approach that we call amortized inference. Amortized inference means that you can do some learning, and hopefully once you've learned something, you can utilize that knowledge somehow instead of trying to estimate things from scratch every time. So this comes in the form of an encoder where we show it noisy images, and we train this encoder to give the pose of the orientation, the unknown orientation of the particle, because there are only so many different orientations we can see this, and 10 million is probably more than we need. So similarly looking images probably have a very similar pose. So we can try to learn this encoder to make that assumption. We build on neural representations, continuous representations of signals, for example, that are parameterized by neural networks to represent these signals. And we do a bunch of other stuff to solve this pretty robustly ab initio, so without making any assumptions on the poses. And this approach has proven pretty effective in comparison to methods like CryoSpark, which is the default method for this. Cryo-AI scales to much larger data sets. It is also better in many cases, and sort of a new approach to solving these challenging inverse problems. If you're a student and you're interested in solving inverse problems or learning to solve them, I'm teaching a class in the winter quarter that's called computational imaging. It's really about solving inverse problems like this, in the large-scale inverse problems, for imaging specifically, though. Okay, so going back to this analogy of the mantis shrimp, I said, hey, there's a sensing mechanism and there's a perception, and so far we've only talked about a subset of the perception, which is the reconstruction. We haven't actually talked about the sensing part, but the really interesting questions in my field, at least over the last few years, have been, how do we really effectively co-design both the sensing, the low-level processing, and also the high-level processing? So reconstruction, I would actually consider a low-level task. It's like you reconstruct some signal from a bunch of noisy measurements. But how do we make decisions based on that, right? So that's what I would call end-to-end optimization. So the question is, how far can you push the ends? On the one hand, you can push it all the way into the instrument, into the detector. On the other hand, you can push it away from low-level reconstruction tasks towards high-level decision tasks. And so the physical image formation model in this case is still going to be the same, but the important part now becomes these parameters theta that parameterize the image formation model, because we might want to optimize them. And then, on the perception side, we might have a reconstruction operator. We might not. We might not have to reconstruct the signal. For medical imaging, maybe we can do a diagnosis directly on the raw measurements that are, captured in some transform domain, or maybe we can classify something based on raw noisy pixels without having to reconstruct it first. But maybe we want to do the reconstruction. The prior is now more important on the perception operator, and perception and reconstruction I sort of distinguish by being a low-level L2 based perhaps reconstruction thing, whereas the perception is maybe a classification task or like a higher level decision task or a planning task, something much more complicated. So we want to jointly optimize all of these components ideally. So that would be the dream, right? And so the questions we can now start to ask are slightly different. They are like, what is the best instrument design for low-level tasks, such as reconstruction, but also for a high-level task? If I'm looking for something specific, how do I design an instrument that is best suited to find this? Where should I make observations to find something? The sky is large. Instruments can capture, at least with high fidelity, only a small part of it. Where should I be looking? How can I use coarse-level information to help guide a finer sampling step, for example? This is a question that we can try to answer in this. Or what is the best algorithm for a given instrument? Maybe two different instruments that do almost the same thing have different algorithms that work better for that specific instrument. So because of this co-design, we can design the whole thing end to end. Okay, so applications here are, for example, going back to this medical imaging example, is where do we place the samples in a CT scanner or in an MRI scanner? How do we design this instrument? What's the exposure time, the radiation time? What is the detector? What's the ideal resolution of the detector, for example? In medical imaging, can we not only reconstruct a volume and then have a radiologist look at the result and tell me what's going on? Can I actually use the AI to tell me if there is a tumor or not? And then if that is possible, what's the best instrument to help me make the best diagnosis? Atomic model fitting, a couple other things. I work on a couple of these different applications, and in particular, this co-design of optics, low-level image processing, and high-level perception. I have a couple of case studies, but we probably don't have enough time to go through all of them. I just want to highlight this idea of monocular depth estimation, where you take a single RGB image and you want to get a per pixel depth map. It's really challenging from a single RGB image because it's a 2D image and you want to get 3D information. But you can use optics to optically encode the point spread function. So you get some defocus blur, basically, that is optimally designed to help the network estimate what the per pixel depth is. And if you look at the very right column and the second from the left column, is we get much better per pixel depth estimation if we use this end-to-end optimization. I'm going to skip one other application that I had planned on talking about because I want to make a different point at the end and not skip over that. So inverse problems is a topic you've probably been exposed to in some level of your career. It's primarily image reconstructions. That's what people think about, at least most of the time. But this idea of end-to-end optimization and co-design of sensing and perception, especially for high-level tasks, I think it's something sort of new and a new capability. But when we think about foundation models for inverse problems, we need to think about different questions even, which is not only how do we make this management better, perhaps, but how do we design entirely new creatures that are better suited for survival, for whatever, in that same environment, right? And for that, we need AI for discovery, not just for reconstruction, not just for classification, but for discovering new things. So the foundation model should help organize knowledge and represent it in a queryable format. It should provide reasoning capabilities, and it should help guide new measurements and instruments, right? But the most important thing is that it needs to help answer lots of important questions. And that's not clear how to use standard methods. So diffusion models is one type of generative model that my group has been working with a lot. You know LLMs and multimodal LLMs. These are slightly different approaches, but that's sort of the knowledge that we offer. You've seen some of this type of effort being rewarded with a Nobel Prize recently is basically AI that helps discovery in science. I'd love to learn more about your problems, and thank you for your attention.
Awesome. Questions for Gordon? Go for it, Raj.
So in your,
you get the mic, please.
Oh yeah. Very cool stuff. So in your cryo, I forget the name, but
Cryo-electron microscope, cryo-EM, yeah.
Example, so you have your like image, and it has like lots of particles in it, like which have these different rotations. How do you go from that sort of big image, which has, let's say, tens of thousands of particles, to knowing that is one particle and that's the second particle?
Yeah, there's sort of standard image processing methods that sort of help you pick the particles. You basically get a small crop around each particle, and it's a fairly well-developed technique. You know, the question then is like, is the particle actually centered, or there's going to be some unknown shift within the crop? It's not going to be centered. Also, not each picked particle is going to be a hit. There's going to be a lot of ones that don't actually contain a structure, right? So, yeah, thinking about that is interesting. We're just using standard methods to do that.
Philip, did you have a question? Okay. Two Philips, go to the mic and take turns.
All right. Thanks very much for going through the cryo here. You know, the 3D molecules seem to be a lot like 3D galaxies, and we make crops, but we call them cutouts or postage stamps, but it's the same kind of thing. So my question was, could you tell us about a case where you were studying a molecule that wasn't very self-similar, but was only somewhat self-similar, kind of like how galaxies are only somewhat self-similar?
So in the context of these molecules, this is usually what we call heterogeneous conformations. So you might have the same sequence, but you know it might be folded in many different ways. So you can think about this as a 3D reconstruction problem or a 4D reconstruction problem. So you actually have different projections of the same molecule, but different incarnations or different instantiations of that same molecule. And then we can either go all the way back to the atomic model and use a physical way to describe the deformation, or what we typically do is a neural network parameterized way, where we say we actually have a 4D signal, and it is parameterized by XYZ, so there is a structure, but it does change. And so we can deal with that as well in some kind of an abstract latent space. It's not always physically interpretable. You can make it physically interpretable or computational efficient. It's not always possible to do both at the same time. But you can do that. What you could also do is I imagine that you don't have the freedom of observing galaxies from many different angles, but perhaps you can observe them at different times and they undergo some physical dynamics that you can describe. So that is virtually also similar to different measurements of the same thing with sort of different projections, right? Or maybe you don't have that, but then you have to make up something. So then maybe you can learn from other galaxies to fill in the missing parts, right? This is really what describes the field of inverse methods. When I describe it to my students, I use the analogy of Jurassic Park. Some of you may have seen the movie. Is you have some of the DNA, but you have to sort of fill in the missing parts with something. And they did it with frog DNA, I think, and bad things happened, right? as we all know. But so hopefully we can do better, right? But so it's a dangerous, slippery slope. You have to be careful about your assumptions and also very explicit about what you're assuming. But I'm sure I would love to chat more and figure out what the exact problem setting is and how to address it.
Let's do one more quick question from Philip.
I'll try to be quick. I'll do my best. About the point that you raised towards the end, I found it very exciting, thinking about the space of getting more foundation perspective on our problem. I wanted to ask, as a community trying to get basically input from all of you, in which direction do you see more of a demand? Like, do we need to define our forward models and how we actually measure these things and this domain knowledge? Is this the critical part, or is it also the question space? Like, where do we care about reasoning?
Oh, yeah.
Which one do you think?
Oh, well, I think there are two aspects. Before you even start sitting down and even thinking about what the foundation model might look like, I think you need to do two things. You need to have a pretty clear understanding of what all the data is, because you basically want to take all the data there is and somehow distill it into this foundation model, right? And that might be visual observations, it might be text, it might be many other things, spectrometer readings, or I don't know, whatever. What is all the world's knowledge that is there that I want to draw from when answering these questions? Because if you leave out one modality, then maybe you can't answer the question well. So you need to find a good way to know what all the sources is. Like all these big foundation models, like Llama and diffusion models and things like that, I mean, they're basically trained on unlimited data. It's scraped from the web, right? So I think you need to think about it this way. Like, what is the combined knowledge of all cosmology today and in the past? And we need to distill all of that in there. Every observation. And then the second question is like, what kinds of questions would you like to ask? Because the design of the model depends on what you're trying to get out of it. If you want to do something like super resolution or hyperspectral reconstruction from RGB images or something like that, some things are much easier to do than others. And so what is it that you're looking for? What are the, is there a constant or some continuous value that you're trying to estimate? Then the foundation model might be different from if you're just asking a question. And so reasoning is not a very well-defined task, right? And there might be benchmarks that need to be done to evaluate reasoning capabilities of existing models or, you know, whatever you're building. So lots of interesting questions, but yeah.
All right, perfect.
Oh.
Let's thank all of our speakers from this session again. And now we will enjoy a break. There are coffee and cookies outside. Encourage you to keep having interesting conversations with others in the room.